now in chapter 7 of our textbook notes, and this is going to be about DNA. And let me make sure I can change the slide. And it's not going to let me change the slide for some reason. Why? Why can I not? There we go. Okay, so by the end of this unit, you should be able to do the bullet points listed, right? So we are going to be able to explain how DNA can be important in criminal investigations, right? Obviously, so that is going to be our forensics, and how DNA evidence is also going to be collected and stored. That's going to be very important. And how crime scene evidence is processed to obtain the DNA. And what is short, we're going to define STR and how that relates to DNA profiling. And how do law enforcement agencies compare new DNA evidence to existing DNA evidence? So then this is a great way for law enforcement agencies to connect crimes. And the use of DNA profiling using what this mtDNA stands for, mitochondrial DNA, and why STRs. And this has to do with family members because mitochondrial DNA is inherited through the mother, and your Y STRs are inherited through the father between the X's and the Y chromosomes. And we're going to be able to compare and contrast a gene, chromosome, intron, and exon. There's a lot of vocabulary in this unit. So here are the vocabulary words in this unit. So you are going to study these vocabulary words, and there is a link on Google Classroom for your Quizlet. And you will also have a Google form quiz on these words. All right, so in the mid-1980s, DNA fingerprinting dramatically changed forensic science and the ability of law enforcement to link perpetrators with the crime scenes. So some very important cases in the 80s, right? We have our O.J. Simpson case, which DNA played a very big role, so it's still relatively new at the time. We also have the case of Colin Pitchfork, which you're going to learn about in your DNA unit project. And also another big case is the Southside Strangler case, right? So those are some pretty big pivotal cases in the 1980s that really brought DNA evidence out to light. And then we have in our early 90s, we have the US military is beginning to collect and store blood samples of soldiers. And this is going to be important in identifying soldiers in times of war. And in 1994, we have the DNA Identification Act. All right, so a little brief timeline of DNA. So as late as 1998, we have the National DNA Index System, which stands for NDIS. And then later on, we then developed what's called CODIS. So you've probably seen or heard CODIS in a forensics themed movie or TV show, NCIS, Bones, things like that. So what CODIS is a computer system that is called Combined DNA Index System that allows for DNA analysis to be stored and compared and then also communicate to law enforcement. So by 2014, look at these big numbers, right? 1.1 million offenders in the DNA profiles, 1.9 million arrestees, and assisted in 246,000 investigations. And that was just by 2014. And by now, you can imagine how large those numbers are. All right, so to go forward in understanding DNA analysis and how it's used in forensics, you have to have an understanding of what exactly is DNA. All right, so DNA stands for deoxyribonucleic acid. Say that 10 times. It's a tongue twister. So what that is it is your genetic material. And it is the genetic material of all living things, right? Plants, animals, flowers, right? All living things are composed of DNA. So with the exception of your red blood cells, all of your cells in your body contain DNA. So the red blood cells do not contain DNA because there is no nucleus in a red blood cell. So in a red blood cell, you're not going to find DNA. So all the DNA found in human cells makes up what's called your genome, right? So your genome is all of the DNA found in your cells. So DNA nucleotide consists of a deoxyribose sugar, and we have what's called a phosphate group, and then here we have our nitrogenous base, right? So this should be familiar to you from living environment. So now we have our nitrogenous base pairs on our double helix, which is the shape of the DNA molecule, right? So you should know that that is the shape of the DNA molecule. And we have A goes with T and G goes with C, right? So A, T, G, C, right? That's going to be important to remember. So you do need to know what two nitrogenous bases pair together. A pairs with T and G pairs with C. So chromosomes, which you have 46 of in your human body, right? So that's 23 pairs of chromosomes. And they're tightly coiled up DNA. So most males, right, there are rare exceptions, have an X and a Y chromosome, right? So that's what makes somebody genetically male, an X and a Y sex chromosome. And females have two X chromosomes. And like I said, I have X because I say that most because there are some genetic conditions where there may be differences. Right, half of your DNA is inherited from your mother. Your mother has two X chromosomes. So your mother is going to contribute your X, right? Notice females have an X and so do males, right? That comes from your mother. And then the other half of your DNA is inherited from your father, which has an X and a Y chromosome. And so if you're a male, you inherit a chromosome from your mother and your X chromosome, and you inherited a Y chromosome from a father. If you're a female, you inherited the X from the father and an X from your mother, and which X comes from which? Well, that I don't know. All right, so that's how inheritance, 50%, right? It's like a 50% lottery, right? All of your genetics is like a random 50% from your mother and your father. So this is a karyotype of those 46 chromosomes, right? We have our 22 homologous pairs of autosomes, right? These are the autosomes, and then followed by the two sex chromosomes, right? So here we have our sex chromosomes. And you notice they vary by size and shape, and they are arranged from the largest to the smallest, right? So from 1 to 22, and this is what's known as a karyotype, right? So this is one of your vocabulary words, and this is one of the activities that you will do in your lab. So a gene is a segment of DNA. So gene and DNA aren't used interchangeably. A gene is just a portion of DNA, and it serves to produce proteins, right? Proteins are a really big and important part of your biochemistry. And genes average about 3,000 base pairs, but they can be composed of many thousands of base pairs. So in your gene, there's regions called coding regions and non-coding regions. So you have exons and introns. So only the exons will be expressed. So your exons are your coding genes, right? Your coding DNA. And your introns are going to be the non-coding. Right? Again, two vocabulary words, exon and intron. Coding, non-coding. So when we collect and preserve DNA evidence, which is what a forensic scientist is going to do, they're going to use the polymerase chain reaction, which stands for PCR, right? Polymerase chain reaction. Trace samples of DNA evidence, right? Trace means very small, right? Super, super, super small, right? Traces of DNA evidence can be amplified, right? Which the word amplified means to be made bigger. And so that we can have enough DNA to test. So that's what PCR does, right? It's a reaction that takes a small sample of DNA and it makes it bigger. And basically, it's by doing that, it's making copies of it. And it's very important to make sure that when collecting evidence and preserving evidence, there's not going to be we want to avoid any contamination, right? This is where gloves and masks, you know me and my technology, right? You're going to have, yep, that's what I did, is because I'm trying to touch the, so you need to have gloves and boot covers, and sometimes even face masks. And when you're storing DNA evidence, it needs to be kept dry and cool during transportation and storage. This is one of the problems that we had in the O.J. Simpson case, right? So if you recall, in the O.J. Simpson case, DNA evidence was packaged in plastic bags and stored in a vehicle in California on a bright, hot, sunny, 90-degree kind of California day, right? So then how you store DNA is very important, because if it's not kept in a cool, dry place, you're going to end up with degradation. You basically want to make sure that you dry a blood sample before storage, and you want to store that in a paper bindle, if you recall making a paper bindle from our beginning of the year lab. So the DNA profiling is the most useful tool for law enforcement since the development of fingerprinting. So fingerprinting is great. It's a individual characteristic, but DNA is kind of like the smoking gun. It is the most important tool, but it's not always necessary. So if you have eyewitness accounts and video accounts and fingerprints and all kinds of other evidence in, a carjacking, for example, you're not going to need to spend the time and resources and money to run a DNA sample if it's not necessary. Because remember, it's also very expensive to run DNA. So scientists use polymorphisms, right? So that's going to be one of our vocabulary words. And this word basically means every single person, except for identical twins, has unique DNA. The DNA is made up of all different characteristics in different coding. So that's what polymorphism is. It's your unique genome that allows you to distinguish one person from another. Now, 99% of all human DNA is the same. So if you look to your neighbor and you look to your siblings and the person that lives across the street and the person sitting next to you in class and the person on TV, 99% of us all have the same DNA. It's only 1% of our DNA that makes us different from one another. And that variability is what allows us to use DNA as an individual forensic identification. And that's called DNA fingerprinting. So the DNA is isolated and it's cut using what's called a restriction enzyme, right? So that's an enzyme that's going to snip the DNA and it's going to create little fragments, right? Little small pieces called restriction fragments, right? The restriction enzyme cuts, making restriction fragments. So each person's length and number of DNA restriction fragments differs, right? This is that 1% difference, right? This is what's going to make you unique, right? All of these little restriction fragments. So then the gel electrophoresis, big complicated word, that was what was used to separate these fragments by size and charge, because the DNA has a negative charge. So now DNA has advanced and the technology has advanced, so we no longer use gel electrophoresis. We use something that's called STR analysis, which is going to analyze shorter and smaller pieces of DNA. So the repeating bases in these STRs, which stands for short tandem repeats, are more easily recovered from often degraded and limited quantities of DNA. This is where also you can pull DNA from a cold case now because of these restriction enzymes and these short tandem repeats. And then again, the Innocence Project, people being set free based on new advances in DNA technology. So the FBI uses 13 core STRs for identifications of Americans. Different countries, the European Union, for example, they use different numbers of STRs. But for all intents and purposes, 13 core STRs is enough to prove uniqueness image law enforcement collecting DNA evidence. Notice taking care with the glove, using the Q-tip, dry, right? Q-tip and gloves to make sure not to contaminate the scene. So where did STRs come from? So a child inherits one allele from each parent for each STR locus. So for each of those 13 STRs, you have inherited one allele from your mother, one allele from your father. And a homozygous genotype occurs when one individual has two alleles that are the same at a specific STR. So if you have two of the same things at that, at STR number 2, then that would be homozygous, right? Because the prefix homo means the same, and the prefix hetero means different. So when you have two different alleles at a particular STR, and we'll have an example coming up of what an STR looks like. All right, so here's some STRs. So here's some chromosomes, and all these little different bands are all the different things that make us unique with the 13 core STRs. And we also have our X and our Y chromosome. So an individual's DNA STR profile is unique, except for identical twins, right? Identical twins are going to have an identical DNA STR profile. A DNA STR analysis is performed using automated machines and computers and fluorescent dyes, and this PCR reaction that we talked about in order to identify. So to talk a little bit about, again, what is an STR? So here we have locus number 9 and 12 and 14 and 15. So if the mother has a 9 and a 12, but the father has a 14 and a 15, so the mother can either donate a 9 or a 12. So then for the offspring, so mother can donate a 9 or mother can donate a 12, right? There's 4 possibilities. So now the father can donate a 14 or a 15. So if I have a 9 with a 14, and I can have a 9 with a 15. Or I can have a 12 with a 14 and a 12 with a 15. So there's 4 possible genotypes in the offspring, right? This is how we can determine the paternity of a child. Same thing. This is just a infographic of what happens in STR typing, right? So here we have our sample. It gets injected into this capillary electrophoresis chamber, right? And it separates things by size. Larger fragments are going to end up down here at the bottom and smaller fragments up here at the top, right? Because smaller is faster. And then we have a laser, right? Then we use some kind of prism and we scatter that light and we make some banding patterns. And then that gets generated by a computer software. And then you get this, which is what we call an STR analysis. So I'm going to skip allele frequency because we're not going to calculate that. So don't worry about that. So here we have a crime scene profile. We have evidence. We have 2 suspects. We have suspect 1 and suspect 2. I need to know, was suspect 1 or suspect 2 present at the crime scene? Which one is going to be our preliminary suspect? Can we eliminate one of these 2 as a suspect? So what we have to do is we have to compare these two suspects STRs. We're basically comparing the graphs and seeing if they match. So here I have a 15 and an 18. So now suspect 2 has a 15 and 18. Suspect 1 only has a 14 and a 15. So 15 matches, but not that 14. And now we have a 17 and an 18. Our evidence is a 17 and a 19. So no 18. So if these 2 things are not present in our evidence, suspect 1 can't be there. And now here we have a 23 and a 24. And here I have a 23 and a 24. So both of these are present, but not these. So suspect 1 is excluded. Now if we look at suspect 2 versus our evidence, we have here an exact match, including the height of the peaks. We have YSTR and mtDNA analysis, which stands for mitochondrial DNA. So again, the Y chromosome, right, is going to be inherited through the male line, right? Because the only males have a Y, genetic males have a Y chromosome to donate. Mitochondrial DNA is passed on only through the mother's line. So now these can only yield class evidence, not individual, because there's just too many possibilities. And now with this kind of DNA, this is where we get into what's called forensic genealogy. So you can have a partial match and you can run these YSTRs and these mitochondrial DNA strands through services like Ancestry.com or 23andMe, and you can get matches. You can get what's called a familial match. And a familial match, right, then you can do forensic genealogy, right, going through birth records and graduation records and church records, and you can literally trace back to a criminal. This is the Garden State Killer is probably the most famous case that was solved through genetic genealogy, and this is bringing in a lot of Fourth Amendment right issues, and I think this is going to be debated for some time to come. All right, so in summary, these are the things that you need to know. DNA is a nucleic acid and it contains the genetic information, and with the nitrogenous bases, right? You need to know which two match with which two. DNA is unique to each individual except identical twins. And the variations with those non-coding parts of the DNA, right, that's the introns, is the basis of forensic identification. And it can help us solve crimes, and it could also help us exonerate falsely accused, right? And this is the Innocence Project. So we can use the PCR amplification, right? You can take small amounts and you can now use that evidence to solve crimes because you're going to be able to make many copies. So DNA contains within its non-coding regions these STRs, and they vary in number amongst individuals, and that's what makes a person unique. That's what makes our DNA unique. So DNA profiling has dramatically improved over the past 25 years, and I'm sure there's still going to be even more improvements as biotechnology, computers, and automated processing becomes more complex and more precise. STR analysis has replaced gel electrophoresis, and I'm sure that there's even more developments ahead in DNA technology in the future. DNA profiling enables us to determine whether DNA samples came from the same person or different people, or can establish kinship, which just means how people are related. Analysis of the base sequences of mitochondrial DNA in the non-coding regions can help identify people through their maternal relatives. And CODIS and NDIS have helped to prevent and solve crimes by improving communication among law enforcement agencies across the country and across the world. And that is it. Thank you. Rewatch